right, let's imagine a scenario where we've got a green dwarf plant that is heterozygous for both traits and a purple tall plant that is homozygous recessive. And I want to know what are the phenotypic ratios that I would see in the offspring. And the first thing we're going to do is we're going to assume that the gene for flower color and the gene for stem height are on different chromosomes, meaning that the traits sort independently. So what do I mean by that? Independent assortment. I mean that if I tell you that the offspring of this cross happens to be purple, do you have any idea as to whether or not they're tall or dwarf? And the answer is no, not if the traits sort independently. It could be purple, dwarf, or it could just as likely be purple, tall. Because knowing the trait for one aspect of the phenotype doesn't give you any insight as to what the other trait is for the other phenotype. The traits sort independently, and the reason they sort independently is this. If they're on different chromosomes, here is the maternal chromosome for the heterozygous organism. This is a germ cell. So this is a cell that will do meiosis. The maternal chromosome and paternal chromosome, they're homologous. They're going to find each other during meiosis 1, and they'll actually be duplicated, right? So let me draw them as duplicated chromosomes. And on the maternal chromosome, we're going to put the dominant allele, and that's going to be present on both of the sister chromatids because they're identical. And then on the paternal chromosome, we're going to put the recessive allele. Now down here, on chromosome number 2, we're going to put the maternal and the paternal. And I'm going to put the dominant allele, again, on the maternal. And I'll put the recessive allele on the paternal. So this is one single germ cell. It goes to do meiosis. So meiosis 1 happens, and we separate the homologous pairs. Meiosis 2 happens, and we would separate the sister chromatids. I'm not going to draw out that process because I expect you to know what meiosis 1 and meiosis 2 looks like. At the end of this germ cell is going to form 4 gametes. 2 of them will have maternal 1 and 2, and 2 of them will have paternal 1 and 2. And this is assuming that crossing over doesn't happen. And the allele combinations are going to look like this. So let's pretend instead of allele combinations, these were like colored marbles. And we'll make this combination red marbles. We'll make this combination blue marbles. But this is not the only combination that can exist. This is one germ cell, right? But this organism here has millions of germ cells, and they're all doing meiosis. And the way that one germ cell does meiosis doesn't influence the way that another germ cell does meiosis. There's another germ cell that, when it goes to do meiosis, happens to line its chromosomes up like this. And so for this germ cell, you're going to get a different combination of alleles in the sex cells. Because meiosis 1 is going to happen, and that's going to separate the homologous pairs. And then meiosis 2 is going to happen, and that's going to separate the sister chromatids. And what we're going to end up with are 4 cells, and 2 of them are going to look like this. They're going to have mom for number 1, chromosome number 1, and dad for chromosome number 2. And then the other 2 are going to have dad for chromosome 1, paternal chromosome number 1, and maternal chromosome number 2. And so that allele combination is going to look like this. 2 of them will have that combination, and 2 of them will have this combination. And so instead of writing out those combinations, let's pretend they're marbles, and let's make this combination be green marbles, and this combination over here be orange marbles. And so because there are millions of germ cells and each one is doing meiosis, and each one does meiosis in one of these two ways, either this combination to produce the red and blue marbles, or this combination to produce the green and orange marbles. When you look at the total number of gametes in the gamete bucket, this is our gamete bucket. In human females, we would call the gamete bucket the ovary. In human males, we would call the gamete bucket the testes. You're going to see an equal proportion of all four combinations. And so when this organism goes to reproduce, they're essentially randomly drawing one of their marbles, one of the gametes, out of their gamete bucket, right? And so in this case, they happen to draw the green combination, which corresponds to a capital G, lowercase d combination. And that is the sex cell that they're going to contribute to the mating process. Now for the other parent, they only have one color marble, because look at the genotype of the other parent. Right? It's homozygous recessive. So it doesn't really matter how the chromosomes line up. At the end of the day, there's only one possible combination. Right? All of the sex cells are going to carry little g, little d. So really what's influencing the phenotype, what is the biggest determination, or the largest determining factor for what the offspring looks like, comes down to this parent right here, right? Because they're either going to donate a green marble, or a blue marble, or a red marble, or an orange marble, right? And each one of these colors is associated with a different combination. But the point is that each color has an equal probability of being selected. Each color has a 25% chance of being selected. And so if they have enough children, we should see 25% of the children made with this fertilization event, 25% of the children were made with this fertilization event, 25% of the children were made with this fertilization event, and 25% of the children were made with this fertilization event, right? And so if you go and look at what all of the possible phenotypes are, you could have this coming from the heterozygous parent. You could have this coming from the heterozygous parent. You could have this coming from the heterozygous parent. Or you could have this coming from the heterozygous parent. The homozygous recessive parent is always going to be sending little g, little d, little g, little d, little g, little d, little g, little d. And so the phenotypes for the offspring, there's going to be 4 different phenotypes, and they're going to be equally represented. You're going to have a green dwarf phenotype. You're going to have a purple, tall, and we would refer to these as the parental phenotypes, right? Because these organisms right here look like this parent, and these organisms right here look like this parent. And then you're going to have phenotypes that are sort of blends between the parents. You're going to have a green, tall, right? So it's going to look like one parent for one trait and another parent for another trait. And then you're going to have purple, dwarf. And again, you're going to see that in a 1 to 1 to 1 to 1 ratio. But what if you don't see that in a 1 to 1 to 1 to 1 ratio? What if, I don't know, instead you see, if they have 1,000 babies, you see 500 babies that look like this, 500 babies that look like this, and then 0 and 0. Right, so instead of seeing 250 across the board, you see 500, 500, 0, 0. Okay, so there's a couple possibilities. Possibility number one, these babies get made, but for whatever reason, they're never born, right? The embryo or the zygote self-destructs well before the baby has an opportunity to be born. That's one possibility. Another possibility is that the heterozygous parent simply isn't making gametes with these combinations, right? So in this case, it would be like the heterozygous parent prefers making the red and the blue, but for whatever reason doesn't want to make the green and the orange combinations, right? But if you've got millions and millions and millions of germ cells, and any germ cell could line up the chromosomes like this or like this, and it's a coin toss, then there should definitely be some of these sex cells. Not only some, there should be approximately half of the sex cells should look like this. Right, but we're not seeing that. So what could explain why none of the sex cells look like this? And the explanation is that we're going to put uppercase D with uppercase G. We're going to put lowercase d with lowercase g. So in this scenario, there is no way to get a dominant G and a recessive D in the same sex cell, right? Because it doesn't matter if I flip-flop the chromosomes, right? I could put the paternal on the left and the maternal on the right, or I could go back to the original configuration. It doesn't matter, right? At the end of meiosis, what you're going to end up with are two sex cells with dominant for both and two sex cells with recessive for both. And so if that's the case, then we go down here to this sex bucket and we have to change the configuration, right? We have to basically get rid of, let me pause it and get rid of these. Right, so I just paused it and erased all of the green, all of the orange, because those combinations simply cannot get made. And so in this case, when these organisms go to reproduce, half the time, the heterozygous organism is going to select a red marble, right? So that would be equivalent to dominant for both. And then the other half the time, the organism is going to select the blue marble, which is equivalent to recessive for both. And so in this case, you're either going to get this fertilization event or this fertilization event. But there's no way to produce an organism that looks like this, and there's no way to produce an organism that looks like this, because the combination, this combination in the sex cells of the heterozygous parent simply cannot exist. Now the reality is under this scenario, what we would actually see is something more like this. Instead of 500, 500, 0, 0, it might be something like 450 if they make 1,000 babies, 450, 450, 50, 50. Which again is interesting because if I put the dominant alleles together and the recessive alleles together, then there's no way I should ever get an organism, an offspring with this phenotype or this phenotype, unless the heterozygous parent makes these combinations some of the time. And how is that possible? And the answer is crossing over. Right, so I'll go over that in another video, but crossing over explains why you get these combinations sometimes. But at the end of the day, if you're not seeing a 1-to-1 to 1-to-1 with this cross specifically, that is overwhelming evidence that the genes are located on the same chromosome. And sometimes we're going to see two phenotypes where the numbers are very, very low compared to the other two, like in this case. Other times it won't be as dramatic. It might be something like 300, 300, 200, 200, right? So instead of 250, 250, 250, 250, it's 300, 300, 200, 200. And this is where we can use our chi-square test to really determine whether or not we're looking at genes that are on the same chromosome or genes that are on different chromosomes. If we assume, this is the observed, if we assume that they're on different chromosomes, then we can write 250. If we assume that they're on different chromosomes, I think that's what I said, they're on different chromosomes, then we can predict 250, 250, 250, 250. And then we can run our chi-square test, we can get our chi-square value, we can compare it to our critical value, and that can help tell us whether or not the genes are on the same chromosome or on different chromosomes. But we'll look at running the chi-square test, we'll look at why sometimes you get numbers that are close to 250, and why other times you get numbers that are nowhere close to 250. Like sometimes you get, like I said earlier, 450, 450, 50, 50. And in some rare cases, you might actually get 500, 500, 0, 0. If the genes are considered completely linked and crossing over, even after accounting for crossing over, you don't get any of these combinations. We do see that sometimes. But that's for another video. That explains the 1-to-1 to 1-to-1 and what it means when we don't see it.